Hi, my name is Sherry Hominy. In this video, I'll walk you through the photosynthesis and cellular respiration LabXchange simulation, and I'll share with you some tips and tricks to support you in using this simulation with your students. So let's get started. The context of this simulation is an investigation that explores the occurrence of photosynthesis and cellular respiration in the light and in the dark. The simulation provides some nice summary graphics that I think are really clear. That plants perform both photosynthesis and cellular respiration is emphasized, which is, I think, very helpful in counteracting the common misconception that many students have that plants perform photosynthesis and animals perform cellular respiration. Asking your students why plants are used in this investigation may help reinforce their understanding that plants do both photosynthesis and cellular respiration. These processes are complementary to one another, with cellular respiration generating carbon dioxide and photosynthesis consuming carbon dioxide. So measuring photosynthesis and cellular respiration in this investigation is based on levels of carbon dioxide. In aquatic systems, CO2 reacts with water to form carbonic acid. Has a higher hydrogen ion concentration and a low pH. Bromothymol blue, which is a colorimetric indicator, can be used to indicate pH as a result of that, the presence or absence of that carbonic acid. So bromothymol is blue at low levels of CO2. With less CO2, there's less carbonic acid formed, and the pH is higher. At a neutral pH, bromothymol blue is green. At a moderate pH, and bromothymol blue turns yellow when there are higher levels of CO2 and more CO2 available to react with water to form carbonic acid. Increase the proton concentration, the hydrogen ion concentration, and lower the pH. Students test their understanding of this relationship between bromothymol blue and CO2 concentrations by reviewing these nice graphics, and then exploring by using the slider that enables them to see that in this test tube, there's a solution of water and bromothymol blue, and CO2 is being added to the solution. And so over time, the more CO2 that's added, the more carbonic acid forms. And we move the slider over for a longer duration of time. You can see that the color change indicates a drop in pH over time, and a nice graph depiction of it here. So students can explore that relationship on their own. And then you'll see throughout the simulation, there are these single choice questions, which are a great way for students to test their understanding as they move through the simulation. One thing I really like about these questions is that students are given feedback regarding their answer, whether they got it right or wrong, and some additional information, and they have multiple opportunities to get the right answer and receive feedback. So for example, here, it says, let's assume that you have a bromothymol blue solution at pH 7, indicated by the green color. Predict what color the solution will turn over time as you blow carbon dioxide into the test tube. So students should know from the previous part of the simulation that increasing the CO2 increases the carbonic acid, which lowers the pH, and that should be indicated by a color change to yellow by the bromothymol blue. If they choose an incorrect answer, for example, they say bromothymol blue turns blue, they'll get the feedback when they submit their answer that, try again, and they'll give a little feedback, and you can see that the students have a few more attempts to get the right answer. When they do select the correct answer, they also get feedback explaining why that's the correct answer. So it's a great learning opportunity for students. So the research question posed in this simulation is, in plants, which of these complementary processes, photosynthesis or cellular respiration, prevails in the dark or in the light? And then we'll move on to the next section, which you can see on this sidebar. Each of the sections of the simulation are detailed. We'll move on to the prediction section. In the prediction section, it begins with a little overview of what was just covered in the context, that photosynthesis consumes or uses carbon dioxide and cellular respiration produces carbon dioxide. And that the more carbon dioxide there is in a solution, the higher the concentration of carbonic acid and protons. So students are provided with, again, some of these questions that are a great way for students to test their understanding. So in this first question, it asks students to predict how the concentration of carbonic acid and the pH change when carbon dioxide is consumed during photosynthesis. So students should consider that photosynthesis consumes CO2, therefore there's less CO2, therefore there's less carbonic acid, which means a lower concentration of hydrogen ions, and therefore a higher pH. And if they choose the correct answer and submit, they'll get that feedback. And again, if they choose an incorrect answer, they have several more attempts, and with each choice an explanation. Next question. Now we can monitor changes to the color of the pH indicator bromothymol blue when photosynthesis changes the levels of carbon dioxide. For that purpose, a water plant called Alodia is placed in a glass tube with water and bromothymol blue. Students are asked to then predict the color change of bromothymol blue after 12 hours of shining light on the water plant. So photosynthesis is occurring, the plant is exposed to light. In photosynthesis, CO2 is consumed, therefore there's less carbonic acid formed, lower concentration of hydrogen ions, and therefore a higher pH, so that the color change should be to blue. And select that choice here, submit the answer, and you'll see the feedback. The next question, students are given an experimental design. Looking at the question, do plants perform more photosynthesis or more cellular respiration in the light? And do plants perform more photosynthesis or more cellular respiration in the dark? And here's the experimental setup. There are 4 test tubes, each of the tubes containing water and bromothymol blue. 2 of the test tubes, 3 and 4, containing Alodia. All of the test tubes closed with a stopper. You might ask your students why the stopper, so that they could understand that atmospheric gases could be exchanged if these test tubes weren't stoppered. And then test tubes 2 and 4 are covered with a light shielding box, so only test tubes 1 and 3 are exposed to light. So this question is asking, what are the controls? What are the variables in this experiment? And so students should understand that tubes 1 and 2 are the controls, and tubes 3 and 4 are the actual experimental samples that represent the variables in the experiment. And when they get that correct answer, they'll get that feedback and an explanation, which I think is really helpful. I really love the explanations that are given with the feedback. It's really helpful. In this next question, students are asked to predict the outcome of this experiment. And this is the experiment that is covered in the simulation. And in this prediction, when they make this prediction, they don't get multiple choice and multiple attempts at making a prediction, although they could reset and make another prediction, but they don't get feedback and a chance to get it right. They don't find out if their prediction is right until the results section of this simulation. So the predictions they make here, as you can see here on this sidebar, we'll go through materials, protocol, run the experiment, and see the results. And that's when this prediction will be compared to the actual experimental results and the ideal experimental results. So in analyzing this setup, in test tube 1, there's no Alodia. It is exposed to light, but no photosynthesis or cellular respiration will occur with no living organism in the solution. So that should remain green. Similarly, test tube 2, no cells photosynthesizing or respiring, so should remain green and neutral. Test tube 3 is exposed to light. It contains Alodia. Photosynthesis will take place. CO2 will be consumed. There'll be less carbonic acid, and this tube should turn blue to indicate the higher pH. And test tube 4 should have no photosynthesis because it's in the dark. cellular respiration will occur, producing CO2, which will increase the amount of carbonic acid, and a drop in pH would be indicated by a yellow change. I'm going to indicate incorrect answers here and just sort of randomly choose blue here, yellow here, green here, and maybe blue here, so that you can see what incorrect results look like when we get to the results section. So I'll submit that prediction, and you'll see that students have a chance to reset and try again, but they don't get feedback as to whether or not they're correct. In this next section of predictions, students are introduced to a spectrophotometer. So a spectrophotometer provides a quantitative measure of the bromothymol blue color change using wavelengths of light and color. So light passing through a sample is either absorbed or transmitted in using orange light wavelength of 615. Lighter colors have less absorbance, and so a lower pH would have less absorbance and more transmittance. And then students can use the slider to experiment with. We change the pH, change the color of the bromothymol blue, what happens to the absorbance? So starting at a very low pH, bromothymol blue is yellow, there's very low absorbance, high amount of transmittance, and we move the slider to indicate, come on, little slider. There we go. Different pHs and color change. You can see that there's intermediate absorbance at pH of 7. And then if we move over to pH of 14, higher pH, darker color of bromothymol blue, very high absorbance, little transmittance. So this provides a quantitative measure of the amount of CO2 in a sample. Next, we'll move on to the materials section. In the materials section, it's very simple. Students are introduced to the materials they'll be using in the lab. Equipment they may not be familiar with include the micropipetter and the spectrophotometer. So they're introduced here, but during the next section, in the protocol, they'll be instructed on how to use that equipment. So they're introduced to the micropipette, the micropipette tip box, which as you know, is very important. Aspects of spectrophotometry, the cuvette lid, and the spectrophotometer. So we'll move on to the next section, the protocol, and go through the process. So it's really important that students read through the protocol carefully and follow each of the steps closely and not progress until they have completed each step. So taking time with that step is really important. And during this protocol, they'll set up the control and experimental tubes. They'll then let the experiment run. Then they will transfer samples from the experimental test tubes into the cuvettes for analysis with the spectrophotometer, and then they'll collect the data. So let's get started on this simulation. I love the detail in each of these steps. Really helps support good lab skills in students, and very effective to have students run through this before doing the actual wet lab. They'll be more successful in the actual lab and have a better understanding of what they're doing if they do this simulation first. So first step is to review the experimental poster, which they can refer to anytime during the protocol. A really nice, clear summary of the experimental setup for test tubes, all with water and bromothymol blue, test tubes 3 and 4 containing elodea, test tubes 2 and 4 protected from light, test tubes 1 and 3 exposed to light. So once we have reviewed that poster, you can see that we've moved on and indicating a color change to indicate that we're on to the next step, where we are to remove elodea from the tank and add them to the tubes. And from the experimental poster, we can see that the elodea goes into tubes 3 and 4. And it's a simple drag and drop into the test tube. You know, if a student accidentally adds elodea to a tube that they don't want to add it to, they can easily remove it from that tube by just clicking on the icon. So we've completed that step, and then we're instructed to seal each tube with a rubber stopper. Great question to ask students is why the rubber stopper is necessary. To prevent the exchange of atmospheric gases, CO2 in the air, with the experimental tubes. So tubes are all stoppered, and we're on to the next step. We're directed to place the light blocking boxes over the tubes that will be in the dark. And again, we can refer to the setup poster to remind ourselves that it's tubes 2 and 4 that receive the light blocking boxes. So tube 2 and tube 4. And you can see that that step is completed. We're on to the next step, which instructs us to turn on the LED lamps to start the experiment. So on goes the lamp, on go the lights, and we're directed to let the experiment run for 12 hours. So we'll click here for this time lapse of 12 hours. And when the 12 hours are complete, we finish that step and move on to the next. After 12 hours, we turn off the LED lights and remove the boxes from the tubes. To remove the boxes, it's simply a click on the icon above the box, and the boxes go back to the shelf. And then we're directed to remove the stoppers from the tubes. And again, that's just a quick click on the icon above, and the stoppers are returned to the shelf. Next, we're directed to use this pH scale poster to estimate the pH of the samples. And we can record those estimates in the notebook. To open the notebook, select the pen icon on this task. So opening up the notebook, we'll record our observations. Test tube 1 seems to have a pH of about, I'm going to call it 6.7. Similarly, test tube 2 looks to be about 6.7. Test tube 3, however, pretty dramatic color change there. Looks to be about, let's call it a 7.7. And test tube 4, another significant color change, looks to be about a 6.0 on the pH scale. And we can save that note. Next, we're directed to select the micropipette. And we're directed to set the volume. To set the volume, you can see it's currently set at 200 microliters. We need to set it at 1,000 microliters. So we simply click on the 200, and we're given, a fairly realistic simulation as to how to change the volume. And that is simply to turn that top tube to adjust the volume. And we do that by increasing up to 1,000. And once we hit 1,000, almost there. There we go. At 1,000, we go back and on to the next step. So we have set that volume. Now we're directed to open the pipette tip box and get a fresh tip on our micropipetter and draw up 1,000 microliters of solution from tube 1. So here we go to tube 1, and I love this aspect of it where students are directed to depress the plunger at the top just to the first stop to draw up a sample, and then the micropipette is, taken over here to cuvette 1, and then to release that sample, we hold that plunger down to the second stop, and the sample is released. Getting a fresh tip, eject the old tip, pick up a fresh tip, and repeat with sample 2. And oops, I just am realizing as I'm looking at this, I'm going to wait to pick up that sample because what I neglected to look at is the next step, which is to seal that cuvette. So I need to drag a cuvette lid to cuvette 1, cover that up. And then I can go on to this step to take a sample from test tube 2. And again, depress to the first stop to draw up a sample. And depress to the second stop to release that sample down to the second stop. I'm going to pause here to put on a cuvette lid. I'm going to release the old tip, pick up a fresh tip, pick a sample from test tube 3, again, going to the first stop to draw up a sample, depressing to the second stop to release the sample. I love that repetition. I love that practice for students. Cover the cuvette, ejecting the old tip, picking up a new tip, and taking our final sample from test tube 4, pressing the plunger to the first stop, releasing the sample by pressing to the second stop, covering the cuvette. And again, good to ask students why it might be necessary to cover. Releasing that tip, placing the micropipetter on the rack, and I love that this includes the step, close the pipette tip box. And then we move on to the next. So students are directed to turn on the spectrophotometer by clicking on the gear button and powering on. While we're waiting for it to warm up, we read a little bit about the spectrophotometer, that there is a light source. We're using 650 nanometers in the orange range. So a constant transmittance through the sample. The sample will vary from the 4 different tubes, and a light detector will determine the transmittance or absorbance and what is absorbed or what is transmitted through that sample. And when we're finished with that, we set the spectrophotometer wavelength to 615. So to do that, we use this slider, and you can see here's the wavelength at the top of the green box. It's currently set at 350 nanometers. We're going to drag this slider to 615 up in the orange range. Oops, went a little too far. Hitting 6. Okay, there's 615. Once we've done that, we click done, and we're ready to rock and roll here with the spectrophotometer. So we open the lid of the spectrophotometer. We review how to hold the samples. So important to understand that the cuvette has frosted sides and clear sides, that the clear sides are to allow the transmittance. Oops, let me go back. Allow the transmittance of the light through. So being careful not to touch the clear sides, placing the cuvette correctly, and allowing that light to be transmitted through. So students get an understanding of how to use the spectrophotometer, only touching this frosted side and not the clear side. So once we've done that, we're ready to start using the spectrophotometer. So starting with the blank, we transfer the blank into the spectrophotometer. We close the lid. Gear button, and we're instructed to hit the gear button and to 0 the spectrophotometer, and that allows a reference point for all the other solutions. So the absorbance is set to 0, and now we're ready to run our samples. So we open the lid of the spectrophotometer. To remove the sample, just click on the icon, and we're ready to transfer cuvette 1 into the spectrophotometer. Close the lid, and enter the value here. It's 0.493. We open the spectrophotometer, remove cuvette 1, move on to cuvette 2, and repeat. We record the absorbance here. 503. Remove cuvette 2. Go on to cuvette 3. Close the lid. Record the absorbance, 0.982. Remove cuvette 3. Run cuvette 4. 0.110. And we remove the last cuvette And you can see that we've completed all the steps in our protocol. The next section is the results section, and we'll move on to that next. So for the results section, remember way back under predictions when we went through the predictions section, and in a question, students were asked to predict an outcome, and I sort of randomly chose outcomes. So the predicted would be what the students had entered way back in the prediction section, and they can see how their prediction compares to their actual results, and also compared to the ideal results, what the expected results will be. And there's a series of questions about all of this. So I love that the student's actual prediction is presented here, and then they have a comparison with the actual and also with the ideal. So in the first question, the ideal results show that the pH indicator color in tube 3 turned blue after the experiment. What's the reason for that? So students should know that 3 was exposed to light. It contained the plant, so photosynthesis occurred. Photosynthesis consumes carbon dioxide, therefore less carbon dioxide to interact with water to form carbonic acid, therefore lower hydrogen ion concentration and a higher pH. And in bromothymol blue, a higher pH is a darker color blue. So the correct answer here should be B. In tube 3, the plant is exposed to light and photosynthesis is favored over cell respiration. Carbon dioxide is used up in the water surrounding Elodia. As a result, the pH increases and the pH indicator bromothymol blue changes from green to blue in shade. And again, as in the other questions throughout the simulation, if the student were to choose an incorrect answer, they'd have additional attempts to get to the right answer, and always with really helpful feedback for correct answers and for incorrect answers. On to the next question. The ideal results show that the pH indicator in tube 4 turned yellow after the experiment. What's the reason for this? And so students should be able to understand that in the dark, in tube 4, in the dark, photosynthesis does not occur, but cellular respiration does. Cellular respiration generates carbon dioxide, and therefore it's available to interact with the water to form carbonic acid, thereby lowering the pH, higher hydrogen ion concentration, and a corresponding color change with bromothymol blue turning to yellow. So check that and submit an answer. Next question, considering the ideal results, would you say that in the light, photosynthesis occurs more than cellular respiration? Based on the experimental results, yes, because of that the blue color change that we observed. And the next question, considering the ideal results, would you say that cells perform cellular respiration in the dark? And yes, you could also ask students to elaborate on this. You know, how do they know that? And it's because it will explain here. They could elaborate. Yes, the color change of the bromothymol blue indicates that CO2 did accumulate, and that change in yellow is an indication of that accumulation of CO2. So the next portion here goes on to show a correlation between bromothymol blue color change and pH and absorbance. And again, a nice slider tool so students can play around with this and experiment, and they can see that as they move the slider at different pH colors. So we can see that we've stopped here midway through here, and that is at a pH of 6.7. And at that 6.7, the corresponding absorbance, and all the way through to the higher pH. And standard curve and the best fit line are illustrated here really nicely. And a nice explanation here as to how creating a standard curve allows us to determine the pH of any unknown bromothymol blue solution by first measuring the absorbance of a standard solution of a known pH to create that standard curve, measuring the absorbance of an unknown pH sample, and then matching the absorbance with the pH value on the standard curve. So a nice tool for students to experiment with, and they can go back to this slider and revisit that. A nice illustration here showing this relationship between absorbance and pH. And then finally, another opportunity to explore, dragging the slider to see what pH value matches the absorbance. So changing the absorbance and the corresponding pH. So this provides the data, the absorbance from samples 1, 2, 3, and 4, and students are instructed to use the slider to determine the pH. So an absorbance of 0.49, taking this down to 0.49, there we go. Here's the, oops, had it there. Absorbance of 0.49 is a pH of 6.9, and we can enter that here. Whoops, 6.9. An absorbance of 0.50. Again, that's a pH of 6.9. Absorbance of 0.98. Taking it up to 0.98. pH of 7.9. And absorbance of 0.11. Taking it all the way down to the other end, that yellow color, 0.11. Here we are, pH of 6. So showing the nice quantitative piece here. We submit our answer. And then an open-ended question. Does the quantitative data you obtained from the spectrophotometer confirm your qualitative pH indicator data? And yes, it does. Students can answer here, submit their answer. Feedback, we expect the quantitative measurement of the pH values by spectrophotometry to mirror the visual qualitative evaluation performed earlier. Measuring absorbance of bromothymol blue at different pH allows us to attach numbers to our results, so making a nice quantitative piece. And then students are asked to reflect. Again, some single choice questions and feedback answers. Let's imagine that plants did not perform cellular respiration in the dark. What results would we expect then? So no cellular respiration in the dark. So if a plant were left in the dark, what would we expect the outcome to be? And students can, make their prediction. So if there's no in the dark, no cellular respiration, they should be able to understand that there'd be no CO2 generated, therefore no carbonic acid, and no resulting pH change. So we would expect the tube 4 to remain green. Let's see if this one is correct. Tube 3 blue, tube 4 green. Let's see what it says. And feedback is correct. And again, students have multiple attempts and feedback at each point. Next question, expanding their thinking about photosynthesis and cellular respiration. Where do plants get their biomass, the plant's body, from? Photosynthesis creates carbon molecules that can be turned into biomass. To determine where plants get their biomass, consider where the reactants for photosynthesis come from. This goes back to why to cover the stoppers and so forth in this process, and students should be able to understand that. They pull the CO2 from the air. And finally, taking this to, expanding this to climate change. Atmospheric carbon dioxide is a dangerous greenhouse gas that contributes to global warming. High levels of carbon dioxide in the oceans acidify the water, endangering many marine species. Could having more plants help combat the negative effects of too much carbon dioxide? And hopefully students are able to take from this experiment that yes, plants, while cellular respiration generates carbon dioxide, which can be converted to carbonic acid and lowering the pH, plants consume that CO2, therefore diminishing the carbonic acid. So yes, plants would lower carbon dioxide levels by performing photosynthesis, taking it full circle here, and they get the feedback on that. Finally, some information about some testing about using the SPEC20 or spectrophotometer. It allows us to more accurately measure the pH changes during photosynthesis. To find out how the pH changed during photosynthesis in tube 3, you measured the absorbance endpoint of your sample after 12 hours. This value didn't give you any information about what happened to the pH in tube 3 between the start and the end point of the experiment. So what's going on in between the start and the end point? So thinking about what's happening with the consumption of CO2. Is it a constant consumption? Does it level off? Does it speed up? So students should think about whether it's a linear relationship, asymptotic relationship, or exponential relationship. And this might be a new concept for them, but hopefully they would understand that initially there would be an abundance of CO2, but at some point that would level off. Cellular respiration would continue, so there'd be a continued production of CO2, but not an excess of CO2. So B should be the correct answer here. Submit that and see if we're correct, and yes. Again, another slider option where students can drag the slider to change the duration of time that the light was shining on the plant. So they get to see what happens between the start and the finish of this process. And CO2 levels becoming more constant and leveling off over time. So the sliders are a great way for students to sort of explore and play around with the concepts. Finally, at the end of the simulation, there's a really nice summary that breaks us down into, I think, about 7 different components, that big takeaways for the students. That photosynthesis consumes carbon dioxide, cellular respiration produces it. Carbon dioxide is essential in building carbohydrates. Measuring carbon dioxide allows us to follow photosynthesis and cellular respiration under different conditions. Bromothymol blue is an indicator, and some information about pH. Using a spectrophotometer to quantify results, how to use a standard curve to analyze data, and then a summary of the different tools that were used. So I hope this video was helpful. Again, this is a great tool to use with your students in preparation for performing this lab. I think you'll find this a very helpful simulation for your students.